So in this section, we're going to be covering the angiosperms, which are the flowering plants. And sperm means seed, and angio means to be covered or encased in, and that's a description of the fruit that develops around the seeds. So here, your objectives are to list the defining features of angiosperms, and then to describe the roles that some animals play in the life cycle of angiosperms, and then be able to explain what double fertilization is and what the outcome of that is. So again, this group is collectively called the flowering plants, and here the ovules, which we talked about with gymnosperms, they're going to be enclosed in diploid tissue at the time of pollination, and a modified leaf structure called the carpel is going to cover the seed, and these carpels are what develops into the fruit. So we are covering the angiosperms, and here's the relationship to the gymnosperms and then to the ferns and their allies, and the cladogram there on the bottom right. So if we talk about the carpel and the origin of the carpel, and we think back to some of the other plants we've been studying, the sporangia can develop underneath or on the leaf itself, and the leaves that bear these sporangia would be called sporophylls. So this was a term we brought up before. And so the sporophyll, this is a leaf that has structures that produce cells. So imagine if you had sporangia here, and these would be, say, megasporangia, and they're producing the megaspore. And then the megaspore goes through development to form the gametophyte, and so within there the egg would be produced, and so this is all occurring on a part of a leaf. But then over time the leaf begins to become modified, and so we see that progression here. The leaf begins to fold over, maybe over many generations of a lineage of plants. And eventually you have the entire leaf folded inward. So the sporophyll now becomes an enclosed structure. And this enclosed structure is now what we would call the carpel. And the carpel would have a chamber in there. So you can see the chamber in there. And that chamber is called a locule. And it's within there that those ovules are produced. There's going to be an egg in there that would get fertilized by a sperm, and as with the gymnosperms, the sperm is delivered by way of a pollen grain, and no water is required for that. The sperm will not have flagella. So the angiosperms themselves, they're very abundant today. That wasn't always the case. They evolved, and they evolved at a time when the planet was dominated by ferns, cycads, the cycadophytes, and the conifers. But they became quite successful and are now the dominant land plants today. And some of the features that helped in this is the production of flowers. The flowers helped promote fertilization of the eggs, pollination. They've developed relationships with animals like insects for pollination, broad leaves in these plants to capture more sunlight, and thicker veins for easier, better transport of water and materials up and down the plant. So all of these are features that you don't see in other plants, the other plants we've been studying so far. And so that helps. So the origin of angiosperms is a mystery, and one of the oldest fossils of flowering plant. And you see the fossil there on the right, and a drawing, an artist's drawing of what this plant might have looked like. And it was given the genus Archifructus, which probably translates into something like ancient fruit or ancient fruit bearer. And this fossil is 125 million years old. So this is not likely to be the first angiosperm. And when we look at the flowers didn't have all the parts that we're going to study here. They lacked structures called sepals and petals. You probably heard of flower petals. If you hadn't heard of sepals, we'll define those here in a little bit. So their exact connection to our modern angiosperms is not known. And so we draw it here with a dashed line here. It is definitely a very primitive form of a flowering plant. But the dotted line there is the exact relationship's not known. And furthermore, this is an extinct group here. Then you see some of the other ones here, monocots, and that we'll be studying, the eudicots, and some other groups. And we're going to talk about Ambarella here in a second, which is a very primitive living angiosperm. And that's the genus, which is Ambarella. And this is what the flower looks like. The petals are not very colorful there. There's the sepals there, and then the reproductive structures there in the middle. It's a very, very primitive flowering plant, and it's very interesting that this one does show some horizontal gene transfer, and horizontal gene transfer is when genes of another species or of another origin is transferred directly into another organism that's alive. We talked about that when we were looking at bacteria and viruses and other things. So this can happen, and it's proposed that perhaps a close relationship between maybe a lichen or a moss growing on the surface of another plant might have provided the transfer of genes. And the genes are from mitochondria here, and we see that genes found in whose origin are from mosses got horizontally transferred to flowering plants. And these are seen in that primitive species Ambarella. This is the proposed phylogeny for that transfer, but don't invest too much time in that. It's just a matter of interest that we do see horizontal gene transfer here within these plants. Now, a bit of flower morphology, the structure of the flower, and once we go over the basic structure of the flower, then we'll be able to go over the life cycle of an angiosperm, the basic one. Now, we want to think of the flower itself as just modified leaves, just like the pine cones or the conifers that we studied in the prior section. And in the laboratory, those could be considered modified leaf structures that grew or that have sporangia develop on them. And so, as with all the structures on the shoots, the flower bud is going to develop from a primordium. Primordium is just a very young, immature structure growing on the plant shoot itself, and it's going to develop on a foot on the stem. So we're going to call that the pedicel, which translates into foot, roughly. And all of the parts are going to be stuck on a structure called the receptacle, which is a widened structure at the end of the stem where the flower is located. The flower is going to be organized into groups of modified leaves called whorls. And we're going to start from the outside and work our way in. And so the outermost whorl actually covers the entire flower as a flower bud before it opens up. And they're made of these green, usually they're green, leafy looking like structures, and they're called sepals. And the sepals together are collectively referred to as the calyx. So the actual whorl is made of sepals. So this is a sepal right here, but all of the sepals together are called the calyx. That's the actual name of the whorl. Okay, so the sepals, and then this is the name of the whorl here. Okay, so this would be the parts of the whorl. They're the sepals. And the name of the whorl is the calyx. I don't know why I put an E there. The next row, or the next one inward, are a lot of times they're colorful, not always. Some flowers are not very showy and not very large at all, like grass flowers. You don't really notice them. They're tiny, and it's because they're wind pollinated. But the next one, petals, right? So the petals, but the name of the whorl is going to be called the corolla. And so They don't give the actual name of the whorl there. So it's called the corolla, and that's all of the petals. And then the next whorl on the end is going to be the male part, and they're called the stamens, and the stamens are two parts: the anther where the pollen is produced, and then the filament. So it's the anther where the pollen is produced, the filament, and then combined that's called the stamen. So all of the stamens, the name of the whorl is going to be Androecium. Okay. And so there's the term there. And andros means male. So the pollen is what's going to contain the sperm. And then the innermost whorl is this green-like structure here. It looks like it has a little white end at the little widened area at the end, a little stalk, and then a widened base there at the bottom. All of that is going to be called carpel, all of it. And usually you have either one carpel, or you're going to have many fused carpels together. And here it looks like they just drew a simple carpel, regardless whether it's one or many. So I'll just say carpel, okay, and or carpels for plural. Those are going to be called the gynoecium. And gynos means female. So this is going to be where the ovule is produced, which is the female gametophyte, and within there is where the egg would be. Now, the the carpel itself has three parts, just like the stamen. You had two parts, had anther and filament. So the three parts of the carpel are the stigma at the top, and the stigma is going to be where pollen lands, and then the pollen tube will grow down in there to go deliver the sperm. The stalk would be called the style. And then the bottom widened area is the ovary. So those are the parts of the carpel. And the carpel or carpels combined is called the gynoecium. So the names of the whorl, starting from the outside going in: calyx, which is made of sepals; corolla, which is the petals; androecium, which is the stamens; and then gynoecium. That whorl is made of the female parts of the carpels. So the males, these are the male parts, and this is the female parts. And that would be a complete flower. So this part right here just goes through the carpels. It mentions what the ovary is, the stigma, and the style. Now, earlier, double fertilization was mentioned. And here, both sperm that are going to be ultimately produced within the male gametophyte are going to be used to fertilize. One fertilizes the egg. The other one's going to fertilize another cell found within the female gametophyte that is going to be used to produce food. We didn't see that in the gymnosperms, so that's sort of unique here. So what we're going to do is talk about the life cycle. And when we went back here, we're looking at within the ovary here. You're going to have a little structure that attaches some integuments, and within there is going to be where the ovule is going to be located. The ovule actually includes some of those cells around there. It's within there that you're going to have ultimately the egg produced in there. And so we have to go through the steps of starting with the spore. Spore's got to produce the female gametophyte. The female gametophyte is going to have the structure that produces the egg. Sometimes call it the archegonium, not necessarily used here in these higher plants overall. So let's say we're in that structure there within the ovary. There's the stalk here. You're going to have the integuments, just like we had with the gymnosperms in there. Within there, you're going to have one cell that's differentiated, that is the megaspore mother cell. And sometimes we can call these mother spore cell, spore mother cells, we can call them sporocytes. So this would be the megasporocyte, in shorter name for instead of all of that there. This cell is going to be its diploid, and this is the one that's going to be the one that goes through meiosis. And so you're going to end up with 4 cells. You're also going to have a little opening in the bottom called the micropyle. That was something we also saw in the gymnosperms, so a name that you should be familiar with. And so when we look here at this life cycle, what I did was I just kind of cut out this area here. So we're here in the ovary. We start with our megaspore mother cell, our megaspore mother site, megasporocyte, and we're just going to go through the part where we develop the female gametophyte in there. So we're going to take a look at that here in this structure here. So your megasporocyte, your megaspore mother cell, goes through meiosis, and when it does, you have 4 cells. Each of those cells are going to be haploid. And the benefit to the meiotic events is that you're going to end up with 4 cells that are haploid, and they're going to have different combinations of the sets of chromosomes they have. So here's where that genetic variation enters in there. You also have the crossing over that occurs. Now you can see in the image here, without having to read the description here, 3 of those cells are going to degenerate. Okay, and so that means we're going to be left with 1 megaspore, 1 spore. We call them megaspores here. That 1 spore is going to go through mitosis, so it's going to go through division, and so at this point here we're haploid. Okay, so we're on the haploid side of the life cycle. That cell goes through 2 divisions. It goes through 3 divisions. So that what this 1 cell is going to form 2 cells, and then those 2 cells divide, forming 4, and then 1 more division of all of those forms 8 cells. This is mitosis here. So all of these cells are going to be genetically identical, but they're going to go through some differentiation. So turn on a different set of genes, and you're going to end up with some different cell types. One of those cells is going to become an egg, which is in bluish color there. You're going to have 2 neighboring cells from those divisions that are going to be called synergids. And then you're going to have some cells on the other side, that 3 of them, that are going to be called antipodal. And then you're going to have 2 in the center that are going to be called the polar nuclei cells in there. And those nuclei, there's 2 of them, which would be the 2 other cells. The total is 8 now, so we have 3 in here near the micropyle where the egg is, the egg and the synergids. Then you have the antipodals over here, that's 6, and then 2 in the middle. Those are the polar nuclei. Now, remember, we're going to have double fertilization. When we're looking at gymnosperms, only the egg got fertilized. Well, here, and that's because the pollen for the gymnosperms was carrying 2 sperm cells. Just the other one degenerated. Well, here, both sperm are going to be used, and 1 sperm fertilizes the egg, and the other one, when you fertilize the egg, you're going to get a diploid zygote. Okay. The other sperm is going to combine and fuse with the 2 polar nuclei, and that's going to make a triploid, what's called the endosperm, which literally translates to inside the seed, and that's going to become the food source. So there's the significance of double fertilization. Okay, so and it's with an I, not antipodal, which means on the opposite of the foot over here. So all of that's described here. There's that antipodal, talks about having 8 haploid nuclei or 8 haploid cells overall. Now, the integuments themselves, which are still on the outside here, those integuments are going to end up becoming the seed coat, just like in the gymnosperms. Now, ultimately, once the pollination and fertilization of the egg, the double fertilization occurs, and the walls of the ovary right here, which is the bottom part here, those become the fruit. Okay, so taking a look here at this entire diagram here, this shows that same part here, that same life cycle. And now we're going to go into the development. And by the way, this right here is the entire female gametophyte. Okay. The female gametophyte, which is part of the ovule here, is sometimes called the embryo sac. Okay. This is all that the gametophyte generation is. All of the structure on the outside, those integuments, are from this generation of sporophyte. All of this is happening within the parent plant. Okay, so these are cells from that, and then this is the gametophyte generation housed within it. Okay, so there's your female gametophyte. Okay, so now we got to go into the male gametophyte, which is going to be the pollen grain, just like with the gymnosperm. So here you're going to start with microspore mother cells, or microsporocytes is another way we can write it. It's the same name as this, so they're the same. They're equal. And they're diploid. So each one of those is going to go through meiosis. So we have meiosis occurring here. And that's going to be 2 divisions. So you're going to end up with 4 nuclei, and each one of those becomes a pollen grain, right? Now, the pollen will leave. Now, this is occurring within the anthers here. Okay, so the little anthers that were there on the stamens, we talked about the flower here. So now we're zooming into one of the anthers there. And the anthers have these chambers here where this is occurring. Basically, these anthers are housing the sporangia, where these microsporangia, where the microspores are going to be produced. So here, your microspores are produced, and they are all haploid. And that nucleus is going to divide once. So you're going to end up ultimately with a structure that is the entire gametophyte that just has 2 different nuclei types. Now, they're genetically the same, but they're going to behave differently because they've differentiated. One of them is going to be the generative cell. My mark here. And it's colored red in this image here. So this is one of those pollen grains. They just drew it up bigger here. The other cell type is going to be the tube nucleus, which is responsible for directing the growth of the tube, the pollen tube. When the pollen, so just like a plant, this pollen would germinate, would grow into a larger, develop into, as part of that is all the gametophyte here. Now the generative cell is going to divide once, and it's haploid. It's going to divide once by mitosis, and it's going to produce those 2 sperm that you see here. This is that pollen tube as it germinates. Now, this is what one of those pollen grains looks like, and then this is your model for the pollen. And when the pollen lands on the stigma, that pollen tube begins to grow, and it grows going down the style, down into the chamber there of the ovary, and then delivering the sperm there into the micropyle. So we can see that process going further along right here. And there are your 2 sperm here. The sperm are coming in here. One sperm is going to fertilize the egg right there. And that's going to become your diploid zygote. And the other 2 sperm are going to combine and fuse with those 2 polar nuclei. So you see that happening here. And that's going to make your triploid endosperm. So when the egg is fertilized, you get your zygote. That's one cell that's diploid. Your endosperm is going to be all of this stuff in here. And all of the cells that make up the endosperm now are going to be triploid. So there's, because 3 nuclei combined, each one was haploid, okay? So that's the basics there of how all of this works. Now what happens with the zygote, it divides by mitosis, and while this is occurring here, the seed is developing further, and the walls of the ovary themselves are changing to form the fruit around the seed, okay? You have different fruit types, but that's the basic idea there. Within this seed now, the integuments, which were on the outside and come from the prior sporophyte generation, are going to go through some changes in a way that protects the seed from drying out, and it also prevents any oxygen from getting in or carbon dioxide from going out. So the metabolism of the now embryo stops, so it becomes dormant. Okay, so that's it. Now the seed ultimately will end up somewhere where conditions are favorable, go through germination, and then start the next generation sporophyte. The embryo is the next generation sporophyte. It's just dormant in there. So this is covered here. I went through it with the image here. I just cut out this image. I'm not going to read through the slides here, but we went over how the pollen was produced. Okay, and remember it's this. This is heterosporous, right? So you had your megaspore with your female and your microspores with the male ones, and they're occurring in different locations, right? One in the ovary, the other one in the anthers. Then we went over pollination. Now, what is pollination? Pollination is not the same as fertilization. Pollination is when the pollen is released, just like with the gymnosperms, and then gets transported to the stigma, and that's pollination. Fertilization does not occur until the pollen is finished germinating, growing, and delivering the sperm. When the sperm is delivered, that's when you get fertilization, right? So we hadn't gotten there yet. So this slides here talk about what happens with those two cells, the generative cells responsible for growing the pollen tube, the sperm, and the tube nucleus for the pollen tube. The generative, like to generate the next generation, that would become the two sperm cells. So the double fertilization significance that was already mentioned. Remember, one of the sperm is going to unite with the egg to form the zygote, and that would be your next generation diploid sporophyte. The other sperm is going to unite with the two polar nuclei to give you your 3N, which is your triploid endosperm, which will be the nutrients for the embryo. Okay, so then the seed forms, the fruit forms. In this section, the seed is described, and your learning objectives are to describe 4 ways in which seeds help to ensure the survival of the plant's offspring, which is the embryo inside. List environmental conditions that can lead to seed germination in some plants. So for the angiosperms, the development of the embryo is going to be suspended or put on pause as soon as special tissues or differentiation occurs that produces within the embryo these meristems. These meristems are going to be the places where you're going to find cells that divide and differentiate as the embryo begins to germinate and grow, and then throughout the entire plant there. And cotyledons as well. These cotyledons sometimes are referred to as embryonic leaves. So in the picture here on the right, you can see the cotyledons identified there. On the embryo, like 2 little rabbit ears here. Here's the rest of the body of the plant, and then down here, this would be there's your meristem, meristem for the root. The embryonic root is referred to as the radicle, which you're going to be measuring in your plant experiment. And then over here, here's your shoot apical meristem, and these cells would actively grow to grow the size of the shoots above ground. There's the embryo, and once it gets to the stage here, it's going to arrest and basically stop its metabolism. The seed coat hardens, and then the seed would be located within the fruit that develops from the ovary and sometimes other parts of the flower. So remember that the integuments are going to end up forming that seed coat, and that seed coat is going to enclose the seed with its stored food, which was the endosperm. So there's that same picture of the seed there, and you might want to just make sure you're familiar with overall with the basic parts of the embryo. There includes the apical meristems, which means the meristems at the tip, the root apical and the shoot apical meristems. Those meristems are going to stay. You're going to see these even in the mature plant later. The cotyledon is the embryonic leaves. In one of the groups of plants, flowering plants, we're going to study, the cotyledons actually absorb most of the endosperm and then store it for use by the embryo. In another group, there's going to be structures that just help transfer the endosperm, the nutrients, to the embryo as it develops. So how do the seeds protect the embryo? One of the things is they help to maintain that dormancy by preventing water loss from the seed, and in doing so, if water can't really get out of the seed, then that means oxygen can't diffuse in, because generally oxygen diffuses better across membranes where there's water present. So that's the thing. So we're going to help maintain dormancy. The seed is going to protect the young plant, the embryo, when it's quite vulnerable. The conditions might not be right for it to be germinating, and so it helps maintain that dormancy and protects it during those times. You don't want the seeds to start germinate at any time because it may begin to grow, and then because the environment's not good enough to get it to a good size where it survives the young seedling. The seed also provides food to the embryo. Okay? And the seed itself also helps with the dispersal of the embryo. The embryo is in there, so the seed will get transported to a new location. And usually that's the fruit's job. Now, the importance of the seed coat. That seed coat is going to form once it does, the metabolic activity ceases, so it goes into dormancy. And the germination, again, cannot take place until water and oxygen reach the embryo. So that's important, is the imbibition, they call it. It's like drinking by the absorption of that seed of water. And the dormancy can, sometimes it's short term, from 1 year to the next, from 1 month to the next, depends on the species. In some cases, dormancy, the embryo can stay viable or able to germinate for years and years. And in some cases, some seeds have been found that are thousands of years old and still are able to germinate. So that's amazing. Now, not all seeds can do that, but some species apparently have, given the right conditions, the dormant embryo, and they can still germinate. Now, germination sometimes for a seed requires very special conditions for that to occur. Some seeds need to be dormant over a winter period, and so they require this cold spell before they can germinate. Others need to pass through the gut of an animal that's consumed the fruit, and the acids in the stomach help break down some of the seed coat. And others may need to be exposed to extreme heat of a fire here, like we see with this pine cone here. Some trees evolve in an area where fire is part of the ecology of that ecosystem. And the seeds that are in the pine cone do not get released until the fire heats up the pine cone and then allows the seeds to release, and then they can germinate as well. So very special conditions might be needed for some. In other cases, like the beans and the radish, you just throw them on some water and they begin to germinate quite nicely. So that's why you're going to be using beans in your germination experiments. So in this section, we're going to look at fruits, and the learning objectives for the fruits are to identify the structure from which the fruit develops, and then to distinguish among some different fruit types. This is not going to be a complete list of different fruit types, but you're going to be looking at what defines a berry, a legume, drupe, and samaras. Now, simply define what a fruit is. It's the mature ovaries. And the ovaries, remember, are part of the carpels, right? So the carpels make up the female part of the flower. Now, when the seed is formed during the formation, the flower begins to develop into a fruit. What part of the flower? Usually it's the ovary. Sometimes other parts of the flower also make the fruit, but the main area, one of the main structures is going to be the ovary itself. Now, it's possible in some cases, for fruits to develop without seeds. For example, bananas have been bred to do that over a period of time. The wild banana itself does have seeds here, and in this case, how are you going to get more banana plants? You're going to propagate them asexually. So you can do that with pieces of a plant, and then the right conditions, perhaps some of the right hormones, and you can get an entire new plant to grow. There basically would be clones of the plant you took the tissues from. So looking at, going back to the images you're familiar with, here's that flower again from earlier, and looking at the inside structure here, the wall of the ovary is going to be referred to as the pericarp. That term should be probably somewhere, there it is, pericarp there. And peri itself, the root there, that word, that part of the word means to surround. And then remember that these are the carpels, so this is the surrounding part of the carpel, right? So that's the name makes sense. Now, there's going to be 3 layers. Okay? Exo for the outside, the exocarp. Meso means middle, the mesocarp, and then the endocarp. And those layers are identified here in the drawing there. Here's the outer layer, outermost layer, the middle layer, and then the innermost layer. And then within there you have your chamber within the carpel there. Where you'll find 1 or more ovules. Sometimes the ovary will have 1 ovule, some other times will have several ovules, and that's part of how we define one fruit or another. Now, when we look at the fruit and then the seed itself, within there, there's going to be 3 basically you're going to have 3 generations there. It says genotypes here, but it might be better to say the generations themselves here. And there's some color coding here to what we're talking about here. The fruit itself, the wall of the ovary, and the seed coat are from the sporophyte generation, from the prior generation. So you can see that it's color coded here. It says this prior. Let me just get that mark out of the way. And so this would be in this beige color here. So again, this includes part of the seed plus the fruit itself. That's from the parent sporophyte. Then the purple color stuff are some remnants of the gametophyte generation. So remember what sporophyte and the gametophyte, which was formed within the ovule, the egg was produced there, and so on. And then the other generation that you have in there is the next generation sporophyte, which is colored in blue. That's your embryo. That's the new generation sporophyte. So you have basically 3 generations in one package there. Okay, so that's a way to think about it. This is just that same image, so we'll go past it here. So now we're going to look at some of the different fruit types here. And just looking at this fruit, this is a familiar tomato. Tomato is actually considered a berry. And you can see there's 4 chambers there. And so that tells me with those 4 chambers that this right here, the ovary is actually made of 4 fused carpels. And so in this particular tomato, right? And then you can see within each carpel, within the chamber there, the chamber is going to be called locule, which I mentioned earlier, Locule, yeah, locule, right? And then within there, you're going to have multiple seeds because there was multiple ovules within each ovary. So sometimes an ovary will have 1 ovule that turns into 1 seed from the fruit. And sometimes we'll have many seeds within each one, and that's the case for a berry. So that's key in there. A berry's going to have many seeds. And by the way, this tomato is just a small version of the wild chili plant we have around here called chile piquin or chile del monte. The jalapeno, those are considered berries as well, and so is the chile piquin. And then we have the bean family. The legumes, they're called. The legumes characteristically make a legume fruit. The peas inside of here and the beans that grow inside bean pods, that's the actual seed. The fruit is the pod itself, and the pod here is made of the entire pericarp. So the exocarp, mesocarp, and endocarp all come together and form this dried fruit itself. And so it's a dry fruit, and it splits along both sides to expose this or to release the seeds that would be within there. So that's what defines a legume. Again, there's no fleshy part there for you to like you might associate with other fruits. So to think about it, the seed pod is the fruit. Okay? And then on the left you have a drupe, and this is a peach here. And for the drupe to form, it's only going to have 1 seed within the ovary, and the ovary wall, the pericarp, the exocarp becomes the skin. The mesocarp is going to be that fleshy, fruity part that we consume. And then the endocarp forms the pit. And the pit is this real hard wood, almost wood-like structure on the inside. And then the seed is way within there. So this structure right here is not the seed. The seed is buried within there. And that endocarp was the inner part of the ovary wall. And so example, here the peach, a plum, and cherries are all drupes. And then here's another kind called a samara. So the samara is on the right. These kind of almost look like the pine seeds that are produced in pine cones. However, there's a difference. The seed is actually encased within the pericarp. Okay, so it's a fruit. This was a flowering plant. Now, this pericarp is actually designed as a wing, so like in the case of maples, when these seeds are released, they kind of float down like a butterfly and hopefully catch a little bit of air. Maybe the wind might carry them and help move them out a ways. And then we have aggregate fruits here, and these would be like mulberries, raspberries. I used to climb trees when I was a kid, and we called these, I think mulberry English, they're called moras here. And what they are as aggregate fruit is it's really just many ovaries. And so each one of these would be an ovary here, so it's an aggregate of ovaries overall, okay? And it's considered one fruit. And then we have multiple fruits. And so that's different than this one. This one's just a bunch of ovaries and it makes one fruit. Over here, you're going to have many flowers growing along one stem. So the entire fruit forms around one main stem there. So there's your main stem. Here's your pericarp, which came from one flower. Here's the next flower, and the next flower, and the next flower, making a fruit like the pineapple. So that would be your multiple fruit. Again, there's other kinds of fruit. They're just not mentioned here. This is not a comprehensive study of botany and different fruit types. But they all have one thing in common. They come from a flower. They are flowering plants, angiosperms. Now, what's important about the fruit is that the fruit itself is going to help disperse the seeds within there. And that's not something that we saw in the gymnosperms. This is one of those things that probably led to an advantage and is the reason why flowering plants are dominant today. So what are some methods that fruits may be dispersed? They get eaten by an animal. They don't get crushed by their teeth. They get taken in and then get spread elsewhere. For example, our mockingbirds here like to eat the little chile piquins. They end up digesting away the fleshy part and then the seeds come out somewhere else when the bird is getting rid of waste. And this is why you see them a lot of times growing near fences or around the edges where the roofs were, where the bird is perched, and then release the seeds with its waste product. Sometimes the seeds hitch a ride, so the fruit is specialized for grabbing and catching hold of fur. Some animals will take and bury these caches of seeds and then forget about them, and then the seed ends up germinating there. Some seeds are specialized to be windblown, and others are transported by water. So on the left here is an example of a fruit that's probably going to attract an animal to eat it, so that would carry the seeds there. Here, this burrs here, these stickers or spinas here, are going to catch the fur on an animal and be transported elsewhere. I used to hate these when I was a kid, walking around barefooted, and then they get in your feet, quite painful. And then here it's like a dandelion here. This is the actual fruit itself, and there's the seed is located enclosed within there, within some of that what would have been the pericarp. And then here's a coconut here. It looks like it's already starting to germinate, but the seed itself is specialized for floating in water, allowing water to help transport it somewhere else for that seed dispersal.